Sexual reproduction has 5 key distinguishing features that set it apart from asexual reproduction. First, sexual reproduction involves two parents, each contributing genetic material to their offspring. Second, specialized reproductive cells called gametes are formed through the process of meiosis. Third, genetic recombination occurs where genetic material is shuffled to create unique combinations. Fourth, fertilization brings together the gametes to form a diploid zygote, the first cell of a new organism. Finally, this process produces genetically diverse offspring that differ from both parents. These key features provide important evolutionary advantages that help species adapt and survive over time. Gametogenesis is the biological process that produces specialized reproductive cells called gametes. During gametogenesis, specialized cells in reproductive organs undergo meiosis to produce haploid gametes. These gametes contain half the number of chromosomes compared to normal body cells. Normal body cells are diploid, containing pairs of chromosomes, while gametes are haploid with only one copy of each chromosome. This reduction is essential for maintaining the correct chromosome number when two gametes fuse during fertilization. Now let's compare the two main types of gametogenesis: spermatogenesis, which produces sperm cells, and oogenesis, which produces egg cells. In spermatogenesis, a primary spermatocyte undergoes meiosis to produce 4 equal-sized, functional sperm cells. This process occurs continuously in the testes throughout a male's reproductive life. In contrast, oogenesis produces just one large functional egg cell. The primary oocyte undergoes unequal divisions, with most cytoplasm retained in the developing egg. The smaller cells, called polar bodies, eventually degenerate. There are several key differences between spermatogenesis and oogenesis. Spermatogenesis produces 4 functional sperm cells with equal cytoplasm distribution and occurs continuously throughout life. Oogenesis produces just one functional egg cell with unequal cytoplasm distribution, and in humans, all potential eggs are formed before birth. Both processes are essential for sexual reproduction, each optimized for their specific roles. Sperm cells are designed for mobility, while egg cells provide nutrients for early embryonic development. Meiosis is a specialized cell division process that forms the foundation of sexual reproduction. In a diploid cell, chromosomes exist in homologous pairs, one from each parent. This example shows 4 pairs totaling 8 chromosomes. Meiosis begins with meiosis 1, where homologous chromosomes separate. Before separation, crossing over occurs, exchanging genetic material between chromosomes. During crossing over, sections of DNA are exchanged between maternal and paternal chromosomes, creating new genetic combinations. In meiosis 1, homologous chromosomes separate into 2 daughter cells. The chromosome number is reduced from diploid 2n to haploid n. Meiosis 2 follows, where the sister chromatids separate, creating 4 haploid cells. The random assortment of chromosomes further increases genetic diversity. Meiosis ultimately produces 4 haploid cells, each with a unique genetic makeup. This variation is essential for sexual reproduction. Let's summarize why meiosis is essential for sexual reproduction. First, it reduces chromosome number by half, from diploid to haploid. Second, it introduces genetic variation through crossing over. Third, random chromosome assortment creates even more variation. Fourth, the reduction in chromosome number prevents chromosome doubling during fertilization. Finally, all these mechanisms enable genetic diversity in offspring. Finally, let's visualize how meiosis prevents chromosome doubling during fertilization. Both the egg and sperm are haploid cells, thanks to meiosis. When they combine during fertilization, the resulting zygote has the correct diploid number. Without meiosis, each gamete would be diploid, and the zygote would have double the normal chromosome count, which would be incompatible with life. Fertilization is the process where 2 haploid gametes unite to form a diploid zygote. Both the sperm and egg cells contain half the normal chromosome count, making them haploid cells. During fertilization, the sperm swims toward the egg and attaches to its outer membrane. The sperm then penetrates the egg's outer layer, and its head containing genetic material enters the egg. Once inside, the genetic material from sperm and egg combine, creating a diploid cell with the full chromosome set. This newly formed cell, called a zygote, now contains the diploid number of chromosomes, half from each parent. The egg has mechanisms to prevent multiple sperm from fertilizing it, a condition called polyspermy. The first defense is the fast block, an immediate electrical change in the egg's membrane that happens within seconds of the first sperm entry. The second defense is the slow block. Cortical granules inside the egg release enzymes that modify the egg's outer layer, creating a hardened fertilization membrane. Finally, egg activation triggers metabolic changes that start embryonic development, making the egg unreceptive to additional sperm. In summary, fertilization accomplishes three key outcomes. It restores the diploid chromosome number, combines genetic material from both parents, and activates embryonic development. Sexual reproduction utilizes two major fertilization strategies, external and internal fertilization. Let's compare these two different reproductive strategies. In external fertilization, eggs and sperm are released into the environment, usually water, where fertilization takes place outside the body. In contrast, internal fertilization occurs when sperm is delivered directly into the female reproductive tract, and fertilization happens inside the female's body. In external fertilization, the female releases eggs into water, while males release sperm nearby. This typically happens in aquatic environments. The sperm swim toward the egg, and fertilization occurs in the water. Many eggs and sperm are released to increase chances of successful fertilization. In internal fertilization, sperm is delivered directly into the female's reproductive tract through various mechanisms such as copulation. The sperm then swim through the reproductive tract to reach the egg, where fertilization occurs in a protected environment. External fertilization is common in aquatic animals like fish, amphibians, and many marine invertebrates. For example, salmon release eggs and sperm into the water during spawning. Internal fertilization is found in mammals, reptiles, birds, and many insects. These animals have evolved specialized reproductive organs and behaviors for this strategy. Animals with external fertilization have adapted by producing large numbers of gametes and synchronizing their release. This strategy is highly dependent on water and generally involves lower parental investment. Internal fertilization adaptations include specialized reproductive organs, fewer gametes, and protected embryonic development. This strategy allows animals to thrive in terrestrial environments and often involves higher parental investment. The two strategies have different success rates and trade-offs. External fertilization typically has lower success rates per gamete, while internal fertilization is more efficient but requires more complex adaptations. These different fertilization strategies represent evolutionary adaptations to specific environments and ecological niches, each with its own advantages and challenges. Embryonic development begins immediately after fertilization with the formation of a zygote. When a sperm cell fertilizes an egg cell, they fuse to form a single-celled zygote, combining genetic material from both parents. Next, the zygote undergoes cleavage, a series of rapid cell divisions. These divisions produce more and more cells without increasing the overall size of the embryo. After several rounds of cell division, the embryo forms a blastula. The blastula is a hollow sphere of cells surrounding a fluid-filled cavity called the blastocoel. At this stage, cells begin to differentiate. Gastrulation is a critical phase where the embryo reorganizes into three distinct germ layers. The ectoderm will develop into the nervous system and skin. The mesoderm forms muscles, bones, and the circulatory system. And the endoderm develops into internal organs like the digestive system. Organogenesis is the process of organ development from the three germ layers. As development continues, cells organize into increasingly complex structures, forming organs like the brain, heart, and liver. Throughout embryonic development, genetic information from both parents guides this complex process. The DNA contains instructions for when and how cells should differentiate and form specific structures. This precise genetic control ensures that development follows the correct sequence and timing. Syngamy is the classic form of sexual reproduction. It involves the fusion of specialized reproductive cells called gametes to form a zygote. This process is the predominant form of sexual reproduction in multicellular organisms. There are three main types of syngamy, classified based on the size and mobility of the fusing gametes. Isogamy involves the fusion of gametes that are similar in size and appearance. This type is commonly found in certain algae like Chlamydomonas, where both gametes are motile and identical. Anisogamy features gametes of different sizes, but the difference is not extreme. Brown seaweeds like fucus exhibit anisogamy, with one gamete slightly larger than the other. Oogamy is the most common form in animals and plants, characterized by a large, immobile egg and a small, motile sperm. In humans and most animals, the small sperm must travel to the large egg for fertilization to occur. Evolutionary biologists believe there is a progression from isogamy to anisogamy to oogamy, reflecting increasing specialization of gametes. Sexual reproduction enhances evolution through genetic recombination, which shuffles genes from both parents. Offspring inherit unique combinations of genes from each parent, creating new genetic variations. Beneficial mutations occasionally arise randomly in individuals. These mutations may provide advantages like disease resistance or better resource utilization. In sexual reproduction, beneficial mutations from different individuals can combine in offspring. When environments change, organisms must adapt to survive. Environmental shifts create selection pressure, favoring individuals with advantageous traits. Sexually reproducing populations adapt more quickly because beneficial mutations can combine and spread faster. Natural selection acts on the genetic diversity produced by sexual reproduction. This combination of genetic recombination and natural selection leads to several evolutionary advantages. As a result, sexually reproducing populations can evolve and adapt significantly faster than asexually reproducing ones. Harmful mutations are genetic changes that can impair an organism's function and reduce its chances of survival. In asexual reproduction, organisms produce genetic copies of themselves. When harmful mutations occur, they're passed directly to offspring. Each generation can accumulate new harmful mutations while still carrying all the mutations from previous generations. This irreversible accumulation of harmful mutations is known as Muller's ratchet and can eventually lead to the extinction of asexual lineages. Sexual reproduction offers a powerful mechanism to eliminate harmful mutations through genetic recombination and natural selection. During meiosis, cells divide to form gametes with different combinations of genetic material. Some gametes may receive harmful mutations, while others may be mutation-free. When these gametes combine during fertilization, they create offspring with various combinations of parental genes and their mutations. This genetic shuffling produces a range of offspring, some with multiple harmful mutations, others with few or none at all. Natural selection then acts on this variation. Individuals with fewer harmful mutations are more likely to survive and reproduce, passing on their healthier genetic makeup. Over generations, we can see how these processes affect population health. In sexually reproducing populations, natural selection efficiently removes harmful mutations, maintaining high population health. In contrast, asexually reproducing populations experience a steady decline in health as harmful mutations accumulate over time with no mechanism to remove them, a clear demonstration of Muller's ratchet. This ability to eliminate harmful mutations represents one of the most significant evolutionary advantages of sexual reproduction, helping species maintain their genetic health over long timescales. Sexual reproduction, while offering genetic advantages, comes with significant energy and resource costs for organisms. The production of gametes requires substantial metabolic resources. Males typically produce numerous small sperm, while females invest in fewer but larger, nutrient-rich eggs. Finding suitable mates requires time and energy that could otherwise be used for feeding or avoiding predators. Organisms may travel long distances, reducing their energy reserves and increasing predation risk. Courtship behaviors demand additional energy investment. Males often develop elaborate displays or engage in competitive behaviors that reduce their energy reserves and can lower survival rates. After reproduction, many organisms invest additional resources in parental care. This includes the energetic demands of pregnancy, nursing, protecting offspring, and teaching survival skills. When compared to asexual reproduction, sexual reproduction is significantly less efficient in terms of energy and resources. Asexual organisms don't need to find mates, engage in courtship, or produce specialized gametes. Despite these substantial energy and resource costs, sexual reproduction persists in nature because, in many environments, the genetic advantages outweigh these significant resource disadvantages. In sexual reproduction, there's a significant genetic cost known as the cost of meiosis. A parent organism contains a full set of chromosomes with both maternal and paternal copies. During meiosis, the parent cell divides to form gametes, each containing only half the genetic material. Each gamete receives only 50% of the parent's genes, randomly selected from maternal and paternal chromosomes. Let's compare the genetic contribution in sexual versus asexual reproduction. In sexual reproduction, each parent contributes only 50% of their genetic material to the offspring. In contrast, asexual reproduction allows 100% of the parent's genes to be passed to the offspring. This 50% genetic contribution might seem like a disadvantage, but it offers crucial benefits. Environments change over time, presenting different challenges to organisms. Asexual reproduction creates genetically identical offspring, while sexual reproduction produces diverse offspring. When a challenging environment appears, genetic diversity becomes crucial for survival. In a changing environment, asexual populations may fail entirely, while some genetically diverse individuals from sexual populations can survive. The cost of meiosis is that parents contribute only 50% of their genes to offspring, compared to 100% in asexual reproduction. While this appears disadvantageous from a gene transmission perspective, the genetic diversity created by sexual reproduction provides crucial adaptive advantages. In constantly changing environments, this diversity outweighs the cost of contributing only half your genes to each offspring. Mammals have distinct reproductive strategies that set them apart from other animal groups. In mammals, fertilization occurs internally. Male gametes or sperm are deposited inside the female reproductive tract. The sperm fertilizes the egg, creating a zygote that will develop into an embryo. Mammals utilize a specialized organ called the placenta for embryonic development. The placenta forms a crucial connection between mother and developing embryo. It provides nutrients, oxygen, and removes waste products. Mammals are characterized by viviparity, or live birth. The developing embryo grows inside the mother's body until birth. This reproductive strategy provides many advantages, including protection during development and higher survival rates. A defining characteristic of mammals is the presence of mammary glands, which produce milk to nourish offspring. Mammals also show complex parental care, with extended periods of dependency, allowing offspring to learn essential survival skills. Though mammals share these reproductive features, there's remarkable diversity in gestation periods and offspring development strategies. To summarize, mammals have evolved a remarkably successful reproductive strategy that includes internal fertilization, placental development, live birth, and extended parental care through nursing and social learning. Monotremes represent a fascinating evolutionary transition among mammals. There are only 5 living monotreme species, the platypus and 4 species of echidna, all native to Australia and New Guinea. Monotremes occupy a unique position in evolutionary history. They represent a transitional form between reptiles and therian mammals, marsupials and placentals, having evolved approximately 166 million years ago. Monotremes have a fascinating mix of reptilian and mammalian reproductive features. They maintain reptilian features like egg-laying with leathery shells and a cloaca, a single opening for reproduction and waste. But they also possess mammalian traits including milk production, hair, warm-bloodedness, and a single bone in the lower jaw. Now let's examine the platypus reproductive cycle in detail. The platypus reproductive cycle begins with mating in late winter to spring. After fertilization, eggs develop inside the female for 10 to 12 days. The female then lays 1 to 3 small leathery eggs and incubates them for about 10 days. When young hatch, they are altricial, hairless, and vulnerable. The mother produces milk through specialized patches of skin, not nipples. Platypuses have evolved several unique reproductive adaptations. Female platypuses have 2 ovaries, but remarkably, only the left one is functional. They lack nipples, with milk instead oozing through specialized patches of skin. Platypus eggs are small, measuring just 15 to 18 millimeters in diameter, much smaller than chicken eggs. During incubation, the female curls around her eggs with her tail, maintaining optimal temperature. The hatchlings, called puggles, use an egg tooth to break out of their shells, similar to reptiles. The reproductive system of monotremes holds significant evolutionary importance. They demonstrate a remarkable evolutionary transition, retaining the ancestral reptilian trait of egg-laying while evolving the mammalian innovation of milk production. Monotremes represent the earliest branch of mammalian evolution, having diverged from other mammals approximately 166 million years ago, providing valuable insights into early mammalian development. Marine animals exhibit diverse reproductive strategies adapted to their aquatic environment. Broadcast spawning is a common reproductive strategy where eggs and sperm are released directly into the water. Corals and sea urchins are examples of marine animals that use this external fertilization method. During spawning events, they release their gametes into the water, where fertilization occurs. Many marine species, especially corals, participate in synchronized mass spawning events. These spawning events are triggered by environmental cues such as lunar cycles, seasonal changes, and water temperature. Coral reefs demonstrate spectacular mass spawning, where multiple species release their gametes simultaneously. Unlike broadcast spawning, some marine animals use internal fertilization, where eggs are fertilized inside the female's body. Sharks are a notable example of marine animals that use internal fertilization. Male sharks have specialized claspers for transferring sperm to the female's reproductive tract, where fertilization occurs. After fertilization, many marine animals develop through planktonic larval stages. This development typically progresses from a fertilized egg to early larval forms, which can swim freely in the water column. These larvae are carried by ocean currents, allowing for wide dispersal and colonization of new habitats. Eventually, the larvae settle and metamorphose into adult forms, completing the life cycle. Marine animals have evolved diverse reproductive strategies that are uniquely adapted to their aquatic environments. These reproductive adaptations are crucial for their survival and ecological success in the world's oceans. Reptiles revolutionized terrestrial life with their amniotic egg, which contains several membranes and a protective shell. This specialized egg protects the embryo from drying out, provides nutrition through yolk, and allows complete development away from water. Reptiles exhibit 2 main reproductive strategies: oviparous or egg-laying and viviparous or live birth. Most reptiles are oviparous, including all turtles, crocodilians, and most lizards and snakes. They lay eggs in protected environments like sand, soil, or vegetation. Some reptiles, however, have evolved viviparity. Certain snake and lizard species retain eggs inside the mother's body, where embryos develop before live birth. A fascinating aspect of reptile reproduction is temperature-dependent sex determination, where the incubation temperature of the egg determines the offspring's sex. In many species, like crocodiles and some turtles, eggs incubated at mid-range temperatures develop into males, while higher or lower temperatures produce females. This temperature sensitivity occurs during a critical period of embryonic development. Climate change is a concern, as it may skew sex ratios in wild populations. Reproductive strategies vary across reptile groups. Turtles and tortoises typically bury their eggs in sand or soil. Crocodilians build nests and exhibit maternal care, guarding their eggs. Lizards show diverse strategies, including both egg-laying and live birth. Some lizard species can even reproduce through parthenogenesis, development without fertilization. Snakes typically lay eggs with leathery shells, though some species give live birth. The ancient tuatara has extremely slow development, with eggs taking over a year to hatch. Insects display an extraordinary diversity of reproductive strategies, making them one of the most successful groups of organisms on Earth. Insects have evolved diverse egg-laying strategies. Some simply scatter eggs, while others carefully place them on specific host plants. Many species create protective egg cases, and some have specialized ovipositors for precise egg placement. Metamorphosis is a key feature of insect reproduction. Complete metamorphosis includes distinct egg, larva, pupa, and adult stages. Incomplete metamorphosis skips the pupal stage. Some parasitic insects exhibit hypermetamorphosis with multiple specialized larval forms. Insects have evolved remarkable specialized mating adaptations. Many female insects can store sperm for extended periods, allowing them to fertilize eggs long after mating. Insect mating rituals can be incredibly complex, involving elaborate displays and chemical signaling. Social insects like bees and ants have evolved sophisticated parental care systems within their colonies. Social insects represent the height of complexity in insect reproduction. They maintain colonies with reproductive division of labor, where only certain individuals reproduce while others care for the young and maintain the colony. Let's explore sexual reproduction in flowering plants, focusing on their unique reproductive structures. Flowering plants have specialized male and female reproductive structures that enable sexual reproduction. The stamen is the male reproductive organ, consisting of a filament supporting an anther, which produces pollen grains. Pollen grains contain the male gametes, or sperm cells, which will fertilize the female gametes. The pistil is the female reproductive organ, consisting of the stigma, style, and ovary. Inside the ovary are ovules, which contain the female gametes, or egg cells, waiting to be fertilized. Pollination is the transfer of pollen from the anther to the stigma. Many flowering plants rely on pollinators like bees. When a bee visits a flower, pollen from the anthers sticks to its body. The bee then visits another flower, transferring the pollen to its stigma. This is called cross-pollination. After landing on the stigma, the pollen grain grows a tube down through the style to reach the ovary. Once the pollen tube reaches the ovary, sperm cells travel through it to fertilize the egg cells in the ovules. After fertilization, the ovules develop into seeds, which contain the plant embryo and stored food. The ovary surrounding the seeds develops into a fruit, which aids in seed dispersal. Plants have a unique life cycle that differs from animals, involving an alternation of generations. This cycle alternates between two distinct multicellular forms, the haploid gametophyte and the diploid sporophyte. In the plant life cycle, the sporophyte is the diploid spore-producing phase. Through meiosis, the sporophyte produces haploid spores with half the genetic material. These spores develop into the haploid gametophyte phase. The gametophyte produces gametes, egg and sperm cells, which unite through fertilization. Fertilization restores the diploid state, creating a new sporophyte and completing the cycle. The balance between gametophyte and sporophyte phases varies dramatically across plant groups. In mosses and liverworts, the gametophyte phase is dominant and most visible, while the sporophyte is small and dependent. In ferns, the sporophyte becomes the larger dominant phase, while the gametophyte exists as a small, independent structure. Gymnosperms, such as conifers, have a highly reduced gametophyte phase, with the sporophyte being the main plant we recognize. Finally, in flowering plants, the gametophyte is extremely reduced, consisting of just a few cells within the flower. Let's compare the alternation of generations in plants to the reproductive cycle in animals. In plants, both the haploid gametophyte and diploid sporophyte are multicellular phases. Plants produce spores through meiosis, which develop into gametophytes that then produce gametes. While plant groups show tremendous variation in their life cycles, animal reproduction follows a more consistent pattern. The alternation of generations has significant evolutionary importance in plant life. It allows plants to specialize each phase for different functions, from reproduction to photosynthesis and structural support. We can observe a clear evolutionary trend from gametophyte dominance in early land plants towards sporophyte dominance in more advanced groups. This reproductive innovation remains one of the key adaptations that allowed plants to successfully colonize land. Sexual reproduction has played a crucial role in human evolution. Let's examine how our species has evolved over time, largely driven by the genetic diversity that sexual reproduction provides. Sexual reproduction creates genetic diversity through the combination of genes from two parents. This genetic diversity provided adaptive advantages that allowed humans to survive in various environments, from tropical forests to Arctic tundra. Beyond biological adaptations, human reproduction evolved complex social components. Pair bonding emerged as a reproductive strategy, creating stable relationships for raising offspring. Extended parental care became essential, as human children require many years of learning and development. These adaptations led to complex social structures that further enhanced our species' success. Humans invest significantly more time in raising offspring compared to other species. Sexual reproduction has thus shaped human evolution both biologically and socially, creating the complex species we are today. This understanding of our reproductive history gives us insight into why humans have such unique social behaviors compared to other species.